Okay, thank you all for coming. So I'm a kind of an engineer who turned cell biologist, so I'm going to talk about how connectivity of cells and the mechanical signals regulate stem cell biology. So we've been told to exercise when we are young, right? And then we also told to exercise when we are older. And we're supposed to do all these other exercises in between to stay healthy throughout our life. But before we get to the sort of like what the mechanical signals mean for the stem cells, I would like to start introducing or defining what this LINC complex means. LINC complex stands for linker of nucleoskeleton and cytoskeleton. And really, if you have a cell that's attached to a surface, you're going to have three basic structural elements. You're going to have a nucleus that sits in the middle, which is denser and stiffer than the rest of the cell. And we also have the cell membrane that encloses the cellular structures. And we also have the cytoskeleton that connect these two elements together. So if you look at a single cytoskeletal element, what you are going to see is that this is actually a continuous structure. On the left, you have extracellular matrix. You have integrin signaling integrins that binds to the extracellular matrix, forming these mechanosensitive focal adhesions. And then these focal adhesions kind of sequester actin cytoskeleton. The actin cytoskeleton then goes in and then binds itself to the nuclear surface. At the nuclear surface, actin is connected by a protein called nesprin. This is a giant protein that protrudes out of the nuclear surface. And nesprin is kind of anchored into the nuclear surface through a protein called SUN. And in this way, this protein complex couples the nucleus to the extracellular matrix, hence the name the LINC complex. Again, the nesprin binds to the actin cytoskeleton, and then the SUN binds to the intranuclear lamin AC network, basically coupling the nucleus to the outside world. So I'm going to talk about today how this complex plays a role in mechanosensitivity and signaling of mesenchymal stem cells. So we know that the mechanical signals are important for maintaining the musculoskeletal system. And just like our muscles, our bones are also able to respond to these mechanical challenges. This could be either a positive regulation or a negative regulation. In terms of positive regulation, it's known that the playing arm of tennis players are much stronger compared to its non-playing tennis arm. And we also know that astronauts lose about 2% bone mass in space per month. So it becomes a real challenge bringing the astronauts back to Earth without giving them extensive rehabilitation. So this ability of bone cells or bones to kind of adapt to these mechanical challenges come from bone cells themselves. And if you take a cross-section of a bone, you will see there's a cortical bone, trabecular bone, and a bone marrow. And if you look a closer look, you will see a number of populations that are actually responsive to mechanical challenges. One of them is osteoblasts. They are the cells that actually make bone. We have osteoclasts, they resorb bone, and these two cell populations are connected to each other with a process called remodeling. And in 10 years from now, all the bones in your body is going to be remodeled by this process. We also have osteocytes. They are the osteoblasts that get trapped in the bone matrix. And again, we have the mesenchymal stem cells. So these are the cells, the progenitor cells for bone, muscle, cartilage, and fat. And since they can also differentiate into fat, we have plenty of fat cells in the bone marrow as well. So the topic of the talk today is how mesenchymal stem cells are regulated by mechanical challenges. And it's been known that if you exercise and a healthy lifestyle, your mesenchymal stem cells is going to be osteogenic. And in the converse, if you are not exercising well or you're in space, for example, your mesenchymal stem cell is going to be more adipogenic and less osteogenic. So in laboratory or preclinical models, we use exercise or exercise analogs to kind of look at how these mesenchymal stem cell fate is regulated. Here's a recent study that we published that we show that with the mice exercising with the training wheel about 6 weeks, increases the bone mass and also at the same time decreases the fat in the bone marrow. So in cellular level, to study this, we use sort of these analogs of exercise and give cells these mechanical challenges. And one of them is the low intensity vibration, or I will call them LIV for the rest of the talk. And these are the signals that we apply anywhere between 30 hertz to 100 hertz and about 1 G magnitude, and in both preclinical, clinical, and cell level models, we have shown that the application of vibrations can increase the bone mass and decrease fat. And similarly, high magnitude strain is also another method that we use. We use this anywhere between 0.1 to 1 hertz with the 2% stretch of the membrane, and we also, others have been shown that it increases bone modeling. And when we look at fat differentiation of mesenchymal stem cells in a petri dish, we'll see that both LIV and strain can decrease the adipogenic commitment of mesenchymal stem cells, as shown by the well-known fat markers adiponectin, AP2, and PPAR gamma. So one of the other things that we look at is how these signals are sensed at the cellular level. And one of the things that we have found, as I mentioned earlier, these focal adhesions are these mechanosensitive signaling platforms that exist at the attachment sites of the extracellular matrix. And we have shown that both LIV and strain can activate these focal adhesion kinases at the focal adhesions, and also result in cytoskeletal remodeling of the actin cytoskeleton, evidenced by the activation of ROE, which is the chief mediator of cytoskeletal remodeling in these cells. We also look at how these cytoskeletal changes happening using imaging techniques. Here is a control cell. You see green is the actin cytoskeleton, and blue here is the nucleus. And here is the response to strain, response to LIV. I would like to get your attention to this area where after vibration we have this F-actin accumulation at the perinuclear area. And in response to strain, what we see is that these are these stress fibers that's crossing across the cell. So one of the questions that we wanted to ask was how this connectivity within the cells can change or affect these mechanosensitivity. And we wanted to look at the role of LINC complexes in kind of mediating this. LINC complex is a two-part protein complex. The nesprine binds to the actin cytoskeleton, and some proteins binds to intranuclear lemon network, connecting the nucleus to the outside world. Here's a cell with or without LINC complexes and how they look. In a control cell, again, you see this green actin cytoskeleton, and the nucleus is in the middle. And you might see the actin cytoskeleton crosses over the nucleus and it kind of pushes it down, making it more flat. In a LINC deficient cell, and we accomplished by this siRNA against the SUN protein, effectively disanchoring the nesprine from the nuclear surface, and you see that the nucleus is more spherical now, and it has no actin cytoskeleton connections to it. When we look at the structural parameters of the nucleus, we see that the nuclear height is increased, nuclear area is decreased, but the nuclear volume remains the same. So when we look at these in cells without functional LINC complex, what we have found is that in the absence of the LINC complex, there was no activation of mechanosensitive pathways. We did this for SI-SUN treated cells and also domain-negative cache treated cell, which is the binding protein or binding complex between the SN and nesprine, so effectively doing the same thing. In both of these cases, we found that the LIV signals require a LINC nucleus to kind of respond to these mechanical challenges. So I would like to switch gears a little bit now and talk about how these signaling events or mechanical signaling events affect the cellular signaling to kind of change the cell fate in mesenchymal stem cells. In addition to the FAC signaling that we have just shown you, when we exercise, there's other signaling events that happen in the cell, and one of them is the activation of AKT. And AKT goes ahead and inhibits the GSK3 beta, and as you know, GSK3 beta is a part of this degradation complex that actively degrades beta-catenin. And beta-catenin is the chief mediator of Wnt signaling pathway, which is known to regulate the regeneration and development of musculoskeletal tissues. So when mechanical signals increase the beta-catenin in the cytoplasm, beta-catenin goes in and enters through nuclear pore complexes to activate the gene's targets. When we get old, however, not only our MSCs become more adipogenic, but their mechanosensitivity is also decreased. We don't have to wait for years, however, to have insensitive MSCs. In a pathology case of aging called progeria, patients again show reduced mechanosensitivity and increased adipogenesis. And one of the etiologies of progeria is this misshapen nucleus. Here's a control cell, and here's a progeric cell. And we know now that the problems in the nuclear structure come because of the mutations in the lemon network. But now we are also learning that these LINC complex mutations also play a role in the phenotype of this. So hopefully in this second part of the talk, I will convince you that this connectivity within the cell is kind of important, regulating this with beta-catenin signaling, and give you a little bit information about what I'm trying to do next. Here is the LINC deficient cells and LINC deficient animals, and you see, I showed you before in the presence of SI-SUN, which is a kind of it disrupts the LINC complex. We see an increased adipogenesis, and at the same time, with the LINC deficient mice, we see a decreased bone mass, suggesting that this LINC complex might actually play a role in how beta-catenin signaling is regulated in cells. And we wanted to ask this question. And indeed, what we have found was that in the absence of LINC complex, we have less beta-catenin in the nucleus, and even mechanical and pharmacological inhibition of beta-catenin is inhibited in the absence of these LINC complexes. So again, just for the time brevity here, that we started with this LINC complexes that connect the nucleus to the outside world, and we have shown that the application of mechanical force can increase the FAC activation and can activate ROA. In this way, they actually produce these cytoskeletal structures around the nucleus, perhaps exerting forces to the chromatin directly and change the stem cell fate. And we also shown that these LINC complexes are intimately connected with the nuclear pore complexes and may play a role in the beta-catenin inward transfer. And one of the things that I didn't show you, we also showed that in the absence of the lamin-AC network, LINC complexes still can exert forces on the chromatin and change the adipogenic commitment of mesenchymal stem cells. In this way, we are currently studying how these LINC complexes are regulated mechanically or otherwise, and we think these regulation of LINC complexes or dysregulation of LINC complexes play a role in the cases where we had the reduced mechanosensitivity and beta-catenin signaling, in cases such as aging, bed rest, or microgravity, or we can utilize these mechanical challenges or regulation of LINC complexes in the cases like tissue regeneration as well as in bioreactors. With that, I would like to thank all the funding mechanisms that support our work, and if you have any questions, I will have to answer. Thank you.